right, so hopefully you guys are seeing my notes on the screen right now. Yes? Yes? Anybody? Am I sharing my screen?
Yes.
All right, cool. All right, because I don't want to go halfway through it like I did yesterday with my computer class and then realize I wasn't sharing my screen. All right, so we left off yesterday at the idea of the replication fork. The replication fork is the point in DNA replication where the building of the new DNA is actually happening. This is where it is split apart the old DNA and new DNA is being formed. And we see this kind of pink blob. That is going to be one of our enzymes. And then these two blue blobs are other enzymes. And it looks like these blue blobs are kind of bringing in the new nucleotides. They look like they're building the new DNA. So my guess, even though it's not labeled, is that the blue blobs in this picture are going to be my DNA polymerase. Because again, my polymerase is my builder enzyme. And again, just a reminder, anytime you see ases are enzymes. ASE at the end of the word suggests that it's an enzyme and definitely suggests that it's a protein. All right, so if you are working on your notes like right now, ladies and gentlemen, this is going to be letter B on the third page of the notes where it says replicating the DNA. All right, so again, replication is a fancy schmancy word for the process by which a cell copies or duplicates its DNA. So instead of having to say that whole sentence, the process by which a cell copies or duplicates its DNA, instead we say replication. So one word versus a whole sentence. It's a time saver. Number 2 right there underneath the letter B on page 3, during DNA replication, the two strands are going to separate and then there will be two replication forks are formed. So we can see the DNA, here's our regular DNA, first it unzips and now I can see two replication forks right here in the middle. Because again, I see two of those little Y shapes as a brand new DNA are being formed. Questions or issues? Okay. All right, so this is going to take us through numbers 3, 4, and 5 on our note catcher. Number 3, new bases are added according to the base pair rules. We know this. We've been talking about this for like weeks at this point. For example, if there's adenine, then we add thymine. If there's guanine, then we would add cytosine. All right, so we see, and I think you have this picture somewhere in your notes. We have the original DNA strand, right? We have the replication fork happening here. All right, so we have going on either side where it is unzipping. And then we have these guys here building our new DNA. And then we have two brand new strands of DNA being formed. And then you have these things called free nucleotides. These are just adenine, guanine, cytosine, thymine that are present in the nucleus so that we can make copies. So these nucleotides are added in a way where the entire molecule has been copied. I do want to remind you, in eukaryotic cells, the cells that have nucleuses, like humans, like we have nucleuses in our cells, trees do, cats, dogs, fish, really any multicellular organism. This isn't just happening on one spot of your DNA and going through the entire strand. Remember, we have multiple replication origins. We have multiple replication bubbles, and this is happening in, again, you can see it right here, it's happening in two directions, and that is to speed up the process. And again, each side of our double helix in my DNA is going to serve as a template for a new strand. So, what is a template? A template, ladies and gentlemen, is when you have it all set up and you just have to put your information in it. The example that I always give is like when I talk about doing presentations in class. Sometimes we go into our Microsoft Word or our Google Slides and we want to find like a pretty template. Oh, this one looks really cool or this one's really pretty. And it's all set up for us with all those cool colors and things like that. All we have to do is add the information. Well, that is what these unzipped DNAs are. When they unzip, they become a template. In other words, all the stuff is there. They just have to add the new information. All right, moving on. All right, here is a question for you. Go ahead and unmute yourselves. You can put it in the chat if you prefer. If I have a template strand that has ACT GCA, what is going to be the new what is a new strand, the complementary strand? What is that going to look like? So go ahead, drop in the chat, unmute yourself, just tell me. This is where I'm waiting on somebody to give me an answer.
If this between DNA and RNA, right?
No, this is, look at my screen. You have ACT-GCA. That is the template strand of DNA. I want to know what is the complementary DNA strand that goes with this. Somebody put it in the chat for me. Oh, two people put in the chat. They're saying it is going to be TGA CGT. CGA. Yep, perfect. Awesome. All right, so ladies and gentlemen, there it is. Oh, come on. All right, so again, base pairing rules, ladies and gentlemen. My base pairing rules tells me that A's always match to T's. So my A, the complementary is a T. C, complementary. G, if there's a T, then the complementary has to be an A. G goes to C. C matches with G. A matches with T. Complementary, this is the base pairing rules. If I have the template strand, then the complementary strand has to be created using base pairing rules. All right, so we are now at number 7. The end result is the production of 2 molecules of DNA that are identical to each other. That is the purpose. We want to replicate our DNA. And again, before any cell division happens during interphase, during the S phase of interphase, we have to double, we have to create a copy of the DNA. The DNA has to be replicated because cells don't function without DNA, and the new cell that we're creating has to have a full set of DNA. Each DNA molecule has what we say one original strand and one new strand. This process is called semi-conservative. All right, we are at letter C. All right, I'm going to leave this up for a minute. First things first. Our first main enzyme in DNA replication is helicase. Again, I want you to notice the ASE at the end of this word. ASE always tells me it is a protein, and 90% of the time, those proteins are going to be enzymes. All right, the helicase is basically the unzipper, right? It's kind of like the zipper pull on your jacket or your pants or your pockets or whatever on your backpack. The helicases are a group of enzymes that break the hydrogen bonds between the complementary pair. This thing right here that is at the replication fork kind of splitting apart my DNA, it looks like a zipper pull, and it is helicase. Helicase is the unzipper. It unzips the DNA molecule and it creates the replication forks, right? So we need to now have this Y-shaped area where we are going to be creating new DNA. When the hydrogen bonds are broken, the two strands of DNA molecule unwind. So we break the DNA and we unwind the DNA. By the way, I don't remember if I added it. No, okay. The unwinder is, by the way, a whole nother protein. It kind of looks like a ring, and it's called topoisomerase. That's going to be on a sheet later on. But again, the main one here is helicase. It is the unzipper. Helicases, again, they are enzymes. They they unzip and break apart the old DNA, separating the parent strand and turning them into template strands. I want to take a minute and go back to today's Do Now. And I just want to kind of remind you, in today's Do Now, I had asked you about, I think it was polymerase originally, but the question in today's Do Now had asked, what is the best accessory, something, something, something. And one of the choices was a zipper pull. So if I ask this question again, and I was asking about DNA, or if I was asking about helicase, helicase basically is a zipper pull. It unzips our DNA. All right, issues or questions? All right, moving on then. Talked a little bit about DNA polymerase today already. We are now at the top of page 4. Okay. DNA polymerase is my builder protein. All right. The main function of DNA polymerase is it is a builder protein. All right. It is going to be an enzyme that is also involved in DNA replication. It adds new nucleotides to the existing chain. So it reads the template and it builds the complementary strand. The rate of elongation is about 50 nucleotides per second in human cells. So your DNA polymerase, every second, like 1/1000, that quick, can replace 50 nucleotides on a DNA strand. Again, DNA replication has to happen fairly quickly because your body is constantly making brand new cells. DNA polymerase, also acts as a proofreader. So not only does it place them, but it also reads to make sure that we are making an exact copy. I want to remind you that DNA polymerase is really, really good at building because it's also, checking its work as it goes. Keep in mind, for every 10 billion nucleotides, polymerase maybe makes one mistake. That's insane amounts of accuracy. But that's a good thing because, again, if your DNA isn't accurate, you're going to have a lot of mutations. And even though mutations are really, really cool in the Marvel universe, oh, look, I got powers. In reality, mutations that happen too quickly to a single individual usually results in really poor health outcomes, sometimes cancers, sometimes genetic disorders, and things like that. So you definitely don't want to have a whole lot of mutations happening because your DNA polymerase can't proofread itself. All right, we are going to pause here. I know we're not exactly done with the notes yet, but that's okay. Like I said, I only want to do about 20 minutes of notes because I always like to give you guys a little bit of time to work on your stuff. So let me get out of my screen share. I am going to stop my video.